Hello and welcome to the D3.1 reproduction review video. Today we're going to be going over both the SL and the HL standards. Standard 1 is a pretty simple one. It's the difference between sexual and asexual reproduction. In sexual reproduction, you have your variation, right? Sexual reproduction creates variation because you're getting half your chromosomes from one parent and half from the other. So that variation comes from new gene combinations. It's really probably better phrase is new allele combinations, but that's okay. And so this is good for a changing environment.
My God.
Asexual.
If we just go outside.
Alright, asexual reproduction creates genetically identical offspring. This is beneficial when there is a constant environment. So it means they're well adapted for the constant existing environment. And so those are the two main differences, right? So we got the new versus genetically identical, and then where they're more beneficial for. Alright, so to allow for this sexual reproduction, you first need to do meiosis. So meiosis is the first step, and this is going to create haploid cells. And so it's going to create these cells that have half the chromosomes, and what they're going to do is break up parental combinations of alleles. So you get these new combinations. You have a cell and you split it in half. So you got half, so we call those haploid. Then what that allows you to do is take the two haploid cells and fuse them together, and you get your zygote. So the fusion of these leads to a zygote. And so this is fertilization. So meiosis, we learned about it. It plays a role in this process. Without it, you can't have sexual reproduction. Alright, now, in terms of the gametes, there are male versus female gametes. Male gametes are typically smaller, adapted for moving or traveling, and they typically are equal in size. But they have low energy reserves because they get there and then they're done. Whereas the egg is typically larger and it has lots of energy reserves. And this is true across both plants and animals. And so then this leads to different reproductive strategies. So for males, right, they're trying to spread as many sperm as possible, whereas females typically are going to have one set of eggs that they then care for. So it's a little different in terms of the strategies too. Alright. Standard 4 is our drawings of the male and female reproductive systems. So we have the male reproductive system first. You can do it. There's side view and front view, and it kind of depends on what you're going for. Alright, so for the male reproductive system, you have down below, you have the testes. And then in the middle, you have, if you draw it from the front view, then in the middle you have the penis. And so off of the testes, you have two things. You have the epididymis. And so the epididymis is where sperm matures. The testes is where sperm is created. The epididymis is where it matures. And then that is connected to a tube called the vas deferens. So the vas deferens goes up like this, loops around, and it will connect to the prostate gland. And there's also the seminal vesicles. Okay, so there's seminal vesicle. Seminal vesicle is for secreting fluid like fructose and mucus and prostaglandin. And then looks the same on the other side. Okay. That connects to the prostate gland. The prostate gland secretes an alkaline solution to cancel out the acidity of the vagina. And then this will, we got the vas deferens, we've got the epididymis. Okay, now we need the urethra. And so then the urethra is the tube which will leave in the center of the penis. So those are the different parts of the male reproductive system. Female reproductive system, same standard. Female reproductive system looks something like this. You have your ovary, your oviduct or fallopian tube, Alright, so you start with the ovary. The ovary is where the eggs are. Then you have the fimbriae, which help get the egg into the oviduct or fallopian tube. So the oviduct is the same thing as the fallopian tube. And then up at the top here you have the uterus. Then we have the lining of the uterus. It's called the endometrial layer. Endometrium. You have the cervix, which it blocks the passageway between the uterus and the vagina. And then you have the vagina. So those are the different parts of the female reproductive system. Alright, standard 5. Okay, so 5 is the menstrual cycle. So there's kind of a lot going on with the menstrual cycle. So the menstrual cycle can be split into 2 different cycles. You have first your ovarian cycle, followed by your uterine cycle. So the ovarian cycle starts in the ovary, and FSH acts on a follicle to cause it to develop. It will increase in size and grow, and then it will secrete, the follicle will secrete estrogen. That estrogen will suppress FSH, and this makes it so only one egg matures at a time. Eventually, the estrogen will cause a spike in LH, and LH and FSH are both coming from the pituitary gland, so that LH will then cause ovulation. So LH causes ovulation. So that's when the egg is released from the follicle. Okay, so this one and this one are pituitary, and then estrogen is an ovarian hormone. During the uterine cycle, the follicle becomes the corpus luteum, and the corpus luteum will secrete progesterone. And progesterone maintains the endometrial layer. So then this will slowly degenerate over time, and as it drops, if no fertilization happens, then you'll have menstruation. And so it will slough off as period blood. And that's the menstrual cycle. Alright. Standard 6 is fertilization. SL fertilization. And so the sperm will fuse with the egg, and the nucleus will enter, but only the nucleus. So the tail will be destroyed. So looks something like this. And then everything else will be destroyed. Now that then you have your two nuclei, the membranes dissolve, and it will undergo mitosis, or joint mitosis. So together they'll combine the chromosomes from each of these, and they'll do what's called joint mitosis, and then you'll have two diploid nuclei. 7. In vitro fertilization. In vitro fertilization is a method to get artificially pregnant. And so what you do is first you stop the cycle, then you do lots of FSH to cause superovulation. You harvest the eggs, and then you combine sperm and egg in a petri dish. So that allows you to then have the embryo develop, and then you will implant it for pregnancy. And that's in vitro fertilization. Okay, that's the end of the human stuff for SL. So now we got the plant stuff. Alright, standard 8. On to plants. Still SL though. So plants do sexual reproduction as well. So first you have the production of gametes. And so the ovule is where the eggs will develop, and then the pollen grains typically develop on the anther. Then from there, you have a couple things happen. So step 1 is you need to have pollination. So the pollination is the movement of pollen from one plant to the stigma of another. And so then when it gets there, you have pollen development. And this is where it will grow a tube, the pollen tube, down the stigma, all the way, or down the style, all the way to the ovary. And then the last step is fertilization. So the pollen then sends the nucleus down, and it's very similar to the one we just saw, where the nuclei will fuse, and you'll have your diploid nuclei. Alright, so you can self-fertilize or cross-pollinate based off of this, right? So if the plant has both parts, it can self-fertilize. But it's still sexual reproduction because they're not going to be identical, not if they make it this way. Number 9, you need to be able to draw a flower and label the different parts. Alright, so you have, we'll start with the female parts since they're in the middle, so it's a little easier to do around it. You have the stigma at the top for receiving pollen. The style is the tube. Together we call these the pistil, with the ovary at the bottom, or the ovule. So we have the ovule as the structure, and then the ovary is inside. Okay. And then you typically have like this. So you have the sepals. The sepals cover it when it's developing, so these cover the bud, so it's doesn't get visitors before it's ready. Then we have petals to attract pollinators. And then we can draw in our male parts. So this whole thing is called the stamen. And then at the top we have the anther. This is the pollen producing part. And then the filament is the thing that holds it up. The whole thing is once again called the stamen. So those are the parts of a flower. Okay, cross-pollination. So plants want to cross-pollinate for the most part because that increases genetic diversity. You can do things like mature the parts mature pollen and stigma at different times. You can have separate male and female flowers to lower the odds of self-pollination, and you can own female plants. And so then, in addition, these are like plants. So in addition to the plant adaptations, you can also have like pollinators. And so these are like animal specific. That'll help with cross pollination. So use the wind to increase the okay, so self-pollination is bad, leads to inbreeding, and so you don't want that when you have your plants. And so what you can do instead is you have the ability to prevent self-fertilization by recognizing self and stopping it from happening. And you can do this at different points, right? You can create no pollen tube if it recognizes it itself. You can have the nuclei be destroyed as it's going down the pollen tube. And you can also prevent it from entering the nucleus. The last flower standard is dispersal and germination of seeds. So to disperse, they can be dispersed by the wind, water, or animals. And animals can either eat it or it can get stuck to them. So these are all different ways of moving them around. And so it's very different than pollination, right? So this is the already fertilized one, and you're just dropping off a seed, basically. And so then the other thing is, once the seed gets dropped off, it has to germinate. And so germination is triggered by water entering, typically. And that will cause gibberellin to activate amylase, which causes you to break down starch to glucose so that you can use it for growing. So this is going to mobilize your food reserves and allow you to start developing, and then that will then have the root will come out, and then the stem will come out, and then you'll start to grow your leaves. And then once you've done that, then it can start growing on its own. Okay, that's the end of the SL. HL standards. 13. So the first thing is the gonadotropin releasing hormone. Or GnRH, controls puberty. And what it does is GnRH from the hypothalamus acts on the pituitary gland and causes the pituitary gland to release FSH and LH. These are both gonadotropins, which means they act on the gonads. And so then these two will go act on either the testes or the ovaries, and they will cause the secretion of testosterone or estradiol. And so then these will lead to puberty. And so they cause like the growth of hair, they cause the development of secondary sex characteristics, etc. Acne. Alright. Then we have spermatogenesis and oogenesis. So spermatogenesis and oogenesis. So both of these are going to start with the spermatogonium, or the stem cell, and the oogonium, which is the egg stem cell line. These are going to undergo mitosis, and they're going to create primary spermatocytes or primary oocytes. These will then undergo meiosis 1, to create secondary spermatocytes, and then one secondary oocyte, but you make one. The other one becomes a polar body. This is also in the fetus. For here, it's all after puberty. There's a pause right here until puberty, and then this resumes. So this one happens in the fetus, this one starts at puberty. Now, these will then undergo meiosis 2, and in this one it creates 4 spermatids, which then develop into sperm through differentiation, and in this one it creates an ovum, but just one. This, once again, this is meiosis 2. This one doesn't happen until fertilization. Okay, so this one pauses multiple times, and you get 4 sperm versus 1 ovum. They're very different sizes, and they result in the different amounts. One of the things we didn't talk about, when we learn about fertilization, we skip the HL standard, which is to prevent polyspermy. And so polyspermy is when multiple sperm fertilize the same egg, and that's really bad. And so we have what's called the acrosome reaction. This is how the sperm actually will penetrate the zona pellicula. So the egg has a layer called the zona pellicula that surrounds it. And so the acrosome reaction eats through that zona pellicula and allows the nuclei to enter. When it enters, it triggers the release of cortical granules. And these then cause the cortical reaction. And this causes the zona pellicula to become hardened, and it will not let another sperm through, and it prevents polyspermy. 16. So as the zygote splits, we call it a blastocyst. And so a blastocyst is the ball of cells that looks something like this. It's a hollow ball of cells that will eventually become the embryo. And so you have the part that will implant and become the placenta. So part of it will become the placenta and the umbilical cord, and part of it will become the embryo. And so it forms the placenta, the mom, and it's going to do this in the endometrial layer. So you have the uterus and you have the endometrium. And so it'll stick to this endometrium, and it will then develop there. And so this will become parts of it, and that's how that will work. pregnancy testing. So there's this thing called human chorionic gonadotropin, which is secreted by the embryo, and it's detectable in the urine because it's removed. And so what you do is you use monoclonal, or an antibody basically, that is specific to HCG. And so what it does is it senses the HCG, it binds to it, and it causes the line to show up. You have two lines, one that tells you that the pregnancy test is working at all, and then the other one tells you whether you're pregnant or not. 18. The placenta. So the placenta is often called like a pancake shape, and its whole purpose is to send nutrients to the embryo from the mother. That's not the best drawing I've ever done. Okay, so this is the endometrium, and here's the other side of the placenta. It has the villi to increase surface area, and so you're going to send CO2, urea, and hormones in this direction, and then back to the embryo you'll send oxygen, glucose, amino acids, vitamins, minerals, etc. And so those are going to go in other directions. You can also, unfortunately, send things like alcohol, so you got to be careful, alcohol, mercury, etc. And so this allows the embryo to develop, whereas the mammals that do not have a placenta cannot develop as far. So then they have to crawl into the pouch, right? So our marsupials, they develop then in the pouch instead. All right, 19. Almost done, just 2 standards to go. So there's 2 hormones involved in this process. The first one is progesterone, which maintains the endometrial layer and maintains the pregnancy, and it drops off very late in pregnancy. The other hormone is oxytocin. Oxytocin is responsible for causing contractions, and then contractions cause more oxytocin, and so this is a good example of a positive feedback loop. And so oxytocin is what will actually lead to giving birth. The final standard is 20. 20 is a weird one. It says that HRT may cause coronary heart disease, but actually, wait, we did more studies and it doesn't cause it. So it's all about like, oh, sometimes studies, so it doesn't though, it does not. That's what newer studies said. And so they're like, oops. And so really it was more about a socioeconomic status. And then it was like, oh, well, we lowered it, but then it didn't. And so, yeah, increase, decrease, and then now it's like, oh, maybe it doesn't do anything at all.